Hello, my name is Arnaud Delorme, and this is a series of video comparing some of the mainstream software for processing EEG data. And I'm the author of EEGLAB, and here I'm comparing other software. And obviously, I'm being very fair with all of them.
False.
And so let's get started. Let's get started with Brainstorm. So I went online, downloaded Brainstorm, installed it on my computer, and added it to the path. And now I'm just going to type Brainstorm. And so it's initializing. So I'm going to create a new protocol, because you need to create a protocol to process data in Brainstorm. So here, file, new protocol. I'm going to call it, protocol 04 is fine. And I want to use the default anatomy, and I also want to use here a template channel file because I don't have the scan channels. So it's telling me, okay, this is not optimal, but let's just do it anyway. So we create the protocol, and then once we have the protocol, we create a new subject, which you can call number 1. And then once we have a new subject, we're going to import some data. So we're going to need to go to the second tab, and then here, I think review raw files. And actually this is much better than import EEG MEG, which we'll do later, but we have to select this one here to link to your raw file to process. I don't know what's called review, but anyway, that's the link we have to click here to select our EEGLAB file. And this is one of the EEGLAB files I've pre-selected. This is part of a BIDS study, and my plan was to process the whole BIDS in different software, including Brainstorm and EEGLAB. But here I'm starting just processing one file from this BIDS study. And I give details about this BIDS study in other of the lectures. So let's just import one file here. This is a pre-300 experiment in which subjects hear audible and standard sound, and then also noise sounds. And they basically have to press a button whenever they hear the audible. So it's beep, 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 boop, beep, beep. And the beep is the standard sound, and the boop is the audible sound. It's a little bit slower, but you get the idea. So here I would choose the file, and here it's showing me the electrodes coordinates according to the template. I wish Brainstorm was automatically aligning these, but it doesn't. So actually you have to go here to common file for subject 1, and then if you want to align it, you have to go to MRI registration, edit, and there's a little bit of learning curve here to actually move these points to the very right location, but eventually it's not that hard. So first you move the X and you bring all the data points to the front. Then I want to move them up with the Z. So now I'm moving them all up. Here, this is not perfect, but this is good enough for our application right here. So then let's close, save the changes, and here we have the EEGLAB data file. So if we can visualize it here, and then as you can see we have standard. If we can move this cursor on the bottom and see the different types of stimuli. So the standard and the oddball, they actually all appear right here, all the types of stimulus. So we're going to build a standard processing pipeline to process this file. And to build a pipeline, you actually have to go here. You realize that quickly because there's not much to do here. It's just the color map. And so actually it wasn't too hard to figure out. I just did it by clicking around. And so here you drag the file you want to process, and then here you press run, which is not intuitive because run is you think it's gonna run something, but it's just actually to build your pipeline. And then here, the first thing we would do in our pipeline is pre-process. So remove the DC offsets, remove any offset of the channels, and then you can run it. Here again, it's not super intuitive, but you can go back to the menu to add to that collection. So that's what I'm going to do. I'm not going to run it right away. I'm going to bandpass filter the data. And these are not the defaults here. I put 1 Hertz and no low pass. So just high pass at 1 Hertz. And then I left the default for the rest. And here it's filtering everything. So that's the high pass, and then so here I can go back to DC correction. And then did I want to do anything else? I tried many of the artifact rejection. Actually, I also tried ICA. ICA first didn't converge, and I think that was because I selected the beginning and the end of the data in this artifact by the filter. Also, I didn't do the DC correction. And once I did that, then it worked. But the problem is that it's trying to use EOG and ECG to find the artifactual components, and I don't have such channels here. And so then it doesn't know what to do, and you actually cannot review the components manually. So I didn't know what to do with the ICA components. The other automated artifact rejection there is, so here I guess you can press delete, is SCP and SSP, sorry. And SSP is a method, if you have blinks, for example, it can find more blinks and remove them. I don't know the exact details. It seems very tailored to MEG data more than EEG data. Also, it requires you to mark some of the data, although there's some might be some automated method. I actually found a method to first flag some of the artifacts and then try to detect them with this. It's pretty complex, and here I only spent a couple of hours, so I decided not to use that method because I couldn't get it to work. Actually, I found how to make that method work later. Here in the menu event, you can select detect other artifacts, and here you have low frequency artifact, high frequency artifact. 5 is the least sensitive method, so you run it. This is the report, 0 error. Then you can click on the file, and now you can see these new events, 40 to 204 Hertz. There's 3 such events. And then the trick here is, so they are rejected once you extract the epoch. You don't want any portions of data that has this event, is to rename them with the prefix bad. And you see the color changes here. And so if we go to the data, like for example here, you see bad for 2. So this region is bad. So no epoch will be extracted over this region. So the trick is to change the prefix to bad. And we can do the same for the low frequency artifacts. So in the final pipeline, that's what's being done. Then we have detect bad channels peak to peak. And this one actually working, but you need to extract data epochs. And it's a very simple method based on thresholding. So we'll do that at the end for artifact rejection. So pre-process, we're just going to do DC correction and high pass. So we're just going to run these two. So here, run both processes. And so here we can see the corrected data. So we go back here, EEG, display time series. It's going to show the data here if we go forward. So we see the data. I'm also going to reference the data. I like to average reference the data. And to do that, you just remove this here. So press delete. Go right here. And again, run the new pipeline. Reprocess. It's in standardized, I think, yeah. Re-reference, average reference. I had to type that in, but it remembers your setting, which is a good thing, and also not such a good thing because there's no button to reset to default. So sometimes you enter something and then you want to go back to default, and I wasn't able to find how to do that. I guess if you install Brainstorm, it just goes back to default. So here, so now this is average reference. And then, so once we have that, we want to extract data epoch. So now we go to this data, and now we put import in database. This is already in the database, but for some reason, when you want to extract epoch, you have to go to import in database. And here we can set up the type of epoch we want to extract. So we're going to extract the oddball with responses and the standard. So we can select these two, and then apply cleaning, since we couldn't get that to extract artifacts because we didn't have the right events. I don't think it's going to do anything. Then you can remove the baseline here if you want to in a specific time range. It might be a good idea here to remove the baseline. So here we're ready, so we're just going to import. And yeah, we want to ignore the epoch because maybe it was outside of the range. And then, so we have oddball and responses. We can compute the average and look at these, but before computing the average, what I want to do is I want to extract the bad epoch. And to do that, you would just remove that here. Run, and then this one, detect bad channel peak to peak. And here, I think I put -1 to 100, and that seems to work to reject about 10% of the epochs. And here I reject the entire trial. So that's a threshold-based method. That's the most basic you can do. All the software have it. That's what everybody was using 30 years ago. Now there's more method right here. So here's detecting peak to peak. It's marking some of them as artifacts. And then we're going to do the same for this one. Artifact, peak to peak. And then we're going to create some average and plot the average. We can also mark the bad channels, which I usually do at that stage. Here, good bad channel, edit good bad channels, and then, for example, you can mark all of these. If you don't want them to use them, you can mark them as bad. And I'm going to do that in the script as well. So here we're going to compute the average. And that's the last thing we're going to do. So again, we put it in the pipeline, run in the pipeline, average files, average by group, and then arithmetic average, or arithmetic average plus standard deviation or standard error. I'm just going to compute the arithmetic average. So here's computing the average for oddball, and then we're going to do the one for standard. There's probably a way to do it for all of them at the same time. Actually, in the script, it's possible. So here, average files. And then you can here go for oddballs, for example, display time series. So that's just the average right now. So you can see it's pretty clean. And if we go to 350, for example, oops, and then we can plot. Maybe if we press the left button here, view topography. So it's showing the nice P300 here at 300 milliseconds. I tried selecting a regions also, so it would average over a region, but I don't think it can plot the average of a region here. I think what you have to do is that you have to use the pipeline, and you have to average over time, and then you can plot the scarp topography for average time series. So that's learning Brainstorm in a few hours, and I apologize if I made any mistake. Please, Brainstorm user, forgive me. But here the goal is really to compare pipelines. So I wanted to build an automated pipeline with Brainstorm. And Brainstorm has a really nice feature right here, is that when you want to run a pipeline, if you add a process here, for example, you can go here, you can save your pipeline, but you can also generate an M script. So here, that's, script 5, for example. I already have a script file. And so this is how it looks. These are all the single trial subjects, single trials, and here this is the process that's applied to single trial. And as long as you use the pipeline, it is pretty straightforward to generate this script. The only issue is that here, for example, when I type import in database, that's not going to be in the pipeline because it doesn't go in this window. So there is actually a way to do that in script, but that's not obvious. Importing the file also, when I linked the file, when I did review raw file, this is not in this box either, so it's not going to generate a script. You only generate scripts for items in this box. So you have to import the file by hand, and once you have imported the file by hand, then you can apply a pipeline you've saved or something. And then once you've extracted epoch, you have also to extract epoch by hand here, and then you can apply another pipeline on the epochs. But I really wanted to do something completely automated, so that's what I tried to do, and that's the script I came up with. So I came up with that looking online at the documentation. In particular, there is section 28 about scripting. So Brainstorm scripting. So that's the last one, and it's a long section, so it tells you how to do some scripting. The documentation for Brainstorm for scripting is relatively poor compared to other languages, and one of the issues I have with Brainstorm, and maybe it's because it's GUI based, is that the functions are not documented. So for example, it tells you okay, use this function, BST report, but if you type help BST report, there's just one line, build report for Brainstorm, so it doesn't describe the parameter. So if you want to know the parameters, you have to look inside the functions, and then you have to read the code, and maybe you can understand the parameter. And luckily the functions are very well built and very structured. So if you want to do that, it is possible. But it's not easy, and you need to be a programmer to go to that level of detail. So if you want to do advanced pipeline, you really have to dig hard. And I had to look inside the code whenever I'm calling a function here, for example, extract, here, import in database. I would look here for keywords, and then look in the code, and then I was able to find this function that imports in the database. Same here for importing the data file. So it is possible to do all that, but it is by no way trivial, and I will make my script available for users. So here is the script I came up with. So this is called new script. Brainstorm likes to save new script. First thing it did, so this is from the Brainstorm documentation page, is that you can start Brainstorm without a GUI, and then it's not going to pop up the GUI, but it's going to run in the background. I name a protocol, and this is the data file. So this is just the EEGLAB data file I want to process, and you need to create a protocol. This is actually the Brainstorm path, so there's a function to get you the Brainstorm path. Then I check if it's a protocol, if my protocol already exists, and if it's the exist, I delete it. And then you see deleting a protocol actually means calling the GUI, either whether it's visible or not visible, and deleting the protocol. Then I recreate it, and then I can import my raw EEGLAB data files. This function is not documented anywhere. You won't see it anywhere on the web. I had to search the code to find it, but it works. There is this function returns you the absolute path, but if you give the absolute path to Brainstorm, it's going to crash. So you have to find the relative path. So I find the relative path because my subject is automatically called new subject. So I found the relative path, and then now starting here, this is the kind of script that Brainstorm generates. So here I just copy and paste the script from the pipeline. So this part is convenient. So you see the first thing I do is remove the baseline. That's what I did on the GUI. And so this script was automatically generated. This one I had it by hand, where I set all the bad channels manually. So I just say, okay, these are my bad channels. I don't want to process these. They're not necessarily bad here. They're just not EEG channels. So I set them not to be processed. As I did here in the channel editor. So it's the same as using the channel editor. The alignment of the channel location file with the MRI, I'm sure it's possible to do from the command line, but I didn't try, mostly because there's lots of manual entries. And so I thought I would have to plug in numbers, and also I don't need that for my pipeline. For my pipeline, I just need the ERP for that subject, for the oddball, and the ERP or single trial for that subject for the distractors, for the standard, I mean. Then this is band passing the data, so same as the data online. This one is, I commented, but I found this function actually to detect back segment of data. And there's a different method to detect back segment of data. It seems to be a pretty powerful function that just look for low frequency and high frequency. So low frequency 127 Hertz, high frequency I think 44 to the highest frequency possible, and detects regions which have these high frequencies. And then it marks the data. Unfortunately, first, this takes a little time. And second, when I extracted the epoch, it completely ignored these bad portions of data. So I gave up on this function. Here, this is the one to do average reference. So I compute average reference here. And this one is the one to extract data epoch. So I extract outboard with responses in standard. There's a bunch of options. So here I've extracted the epoch, and then here it's detecting bad epoch. And actually when you do it from the command line, apparently you can just put all the epochs together, and it's going to detect the bad epoch, whether they're standard and oddball. And then I create the average, and again, one call will do both average for both the oddballs and the standard. And this is the average type, so this one is just this regular arithmetic average. Then I set the current time to 0.3, so it was when I click on the window to set my vertical bar, my vertical red bar, and then I plot the scalp topography as I did. And then very importantly, this one also took me quite some time to find, I retrieve the single trial. I retrieve the single trial so I can do more statistics on them, and I can compare between different software. So here I'm just going to run that script. And see it's deleting test EEGLAB, creating a subject, importing the data, extracting standard. The GUI crashed twice on me, so I got some error. Like for example, when I said I tried to import bits, I had a standard bits, but I didn't have MRI, so I think that's why it didn't behave properly. So now it's done extracting the average, and now it's computing the average, and then it's going to plot the results. So here, and obviously this is wrong because I didn't line the electrodes. So the last part I wanted to show you here is a cool feature which is when you use the pipeline, you can actually run some code here, some custom MATLAB code. And apparently you can also create plugins easily. That's documented on their webpage. If you put it in a specific folder, it's going to detect them automatically. But this one is nice to run MATLAB command. So because I have EEGLAB on my path, I created empty. So this passed data and time vector. Unfortunately, it doesn't pass channel information and event information. The data and time is already something. So for example, here I create an empty dataset, and then I create a EEGLAB dataset. I re-reference, and then I export back the data to Brainstorm. So this is referencing within EEGLAB. And so that's it for Brainstorm, generating one file. There's many more features in Brainstorm. Brainstorm is mostly done for source localization, and I didn't use any of these features. Thank you for watching, and please also check the video comparing the pipelines.